Welcome to the Deep Dive, where we unlock the profound insights from groundbreaking research, giving you a shortcut to truly understanding the discoveries that are reshaping our world. Today, we're embarking on a truly remarkable journey, one that takes us deep into the hidden, intricate biological mechanisms that silently govern our health every single second. I mean, how often do you pause to consider the ceaseless, elegant dance happening inside your body at a molecular level? What if we can not only witness those breathtaking interactions with unprecedented clarity, but also understand their precise language, and then, crucially, learn to communicate back, perhaps even, whisper instructions or shout commands to restore health? Imagine a scientist who possesses this almost superhuman ability to literally see the unseen, to translate the invisible choreography of molecules into actionable knowledge, thereby unlocking potential cures and redesigning the very fabric of biological communication. That's the extraordinary individual we're deep diving into today. K. Christopher Garcia, he's not just an American scientist,
He's a structural biologist who holds a distinguished professorship at Stanford University School of Medicine, an investigator of the prestigious Howard Hughes Medical Institute, and a highly respected member of both the National Academies of Science and Medicine. And to immediately underscore the profound translational impact of his visionary work, it's vital to know he's also a co-founder of several pioneering biotechnology companies. This isn't just theory. This is discovery moving directly to application.
Right, and what's truly fascinating here, I think, is that Garcia's entire approach stands as a testament to bridging what often appears to be a chasm between fundamental, curiosity-driven basic science and its direct, impactful translational application. This intersection is not just vital, it's becoming the very crucible of modern biological innovation, really. Our mission today is to meticulously explore how he has masterfully integrated the powerful disciplines of structural biology, biochemistry, and protein engineering. His goal? Well, it's to understand with atomic precision how cell surface receptors function, to reveal the fundamental, often elusive mechanisms that drive human disease, and then, armed with that profound understanding, to design entirely novel therapeutics. And his work is exceptionally broad, spanning critical areas from the intricate world of immunology, which protects us, to the complexities of neurobiology and the very foundations of developmental biology.
it's an expansive and breathtaking spectrum, isn't it?
It really is.
From deciphering the most minute molecular handshake to engineering sophisticated solutions for some of humanity's most complex and challenging diseases. Okay, let's unpack this incredible journey of discovery, delving into how visualizing these impossibly tiny molecular interactions has yielded insights with truly enormous implications for human health and, well, our understanding of life itself. Okay, so to truly appreciate the profound impact of K. Christopher Garcia's contributions, we probably need to start at the beginning, right? Lay the bedrock of his career by examining his foundational education and the unique structural biology lens he meticulously developed. His academic trajectory reveals a consistent, almost relentless pursuit of a fundamental atomic-level biological understanding. He kicked things off at Tulane University, earning his Bachelor of Science in Biochemistry.
A solid chemical and biological grounding there.
Exactly. But it was his graduate work at Johns Hopkins University School of Medicine that really set the trajectory for his future groundbreaking work. He got his PhD in biophysics under El Mario Amsel.
Right, and that early specialization in biophysics is key.
Why so key?
Well, biophysics applies the principles of physics to biological systems. It honed his ability to think about the physical forces, the structures governing life. It wasn't just about what molecules were there, but how they physically interacted, their mechanics. This shaped his talent for visualizing complex structures later on.
Ah, okay, so he was learning the mechanics of life's machinery, essentially.
Precisely. And that strong, almost architectural foundation in biophysics was then, profoundly amplified by his postdoctoral research. He spent a pivotal period at Genentech, working in the labs of David Goodell and Tony Kostiakov.
Genentech, that was a big deal back then, wasn't it? Especially for biotech.
Huge. And this wasn't just any postdoc. It was an immersion into what were genuinely nascent technologies at the time. Things like protein engineering and recombinant protein expression. Think about it, late 80s, early 90s. The ability to rationally design or even just reliably produce specific proteins in large amounts, that was revolutionary.
Right, we sort of take it for granted now.
We do. Genentech was pushing those boundaries. So Garcia's time there was critical. He wasn't just watching, he was developing the tools, the practical methods, and maybe even that sort of entrepreneurial mindset to manipulate and understand proteins on a whole new scale. And following Genentech, he then further solidified his expertise in structural biology. He went to the Scripps Research Institute, working in Ian Wilson's lab.
Another top institution. What was the focus there?
Wilson's lab was a powerhouse in X-ray crystallography, the technique for figuring out the 3D structure of molecules. So this phase really refined his skills in crystallizing tricky proteins and deciphering their atomic structures. So what you see emerging is this extraordinary multidisciplinary training from these top-tier places. And that Gentech experience in particular gave him this truly unique toolkit.
How so?
It blended the deep theoretical structural insights with the very practical hands-on application of protein engineering. This unique fusion, the ability not just to see and understand, but to actively design and build new biological molecules that became a defining hallmark of his lab's work later on.
So he could ask the big questions and then actually engineer the tools to find the answers.
Exactly. And potentially engineer the tools to fix the problems too.
It's undeniably clear how that incredibly diverse and hands-on training laid the intellectual groundwork for his research philosophy at Stanford. So today, his lab there is this vibrant hub, right? A place where structural biology, biochemistry, protein engineering, they're not just existing side by side, they're deeply integrated.
Absolutely. Working in concert.
And the core theme driving it all is understanding how cell surface receptors, the cell's antenna, basically sense cues from their environment.
Picking up those signals, those extracellular ligands.
And then, crucially, how they transmit those signals inside the cell, triggering responses.
Right, the transduction part.
So his main goal is to clarify, like with atomic detail, the structural and mechanistic basis of how these receptors get activated, particularly in systems relevant to human disease.
But it doesn't stop at just understanding.
No, that's the key part, isn't it? Once that deep knowledge is there, the rules of communication are figured out, that information is then strategically exploited.
To design and engineer completely new molecules.
Tailor-made with therapeutic properties.
Precisely.
So for you listening, what does this all mean? Well, think of your cells constantly listening to their surroundings, processing signals every second, deciding to grow, move, whatever. Garcia's work is like being a master cryptographer.
Deciphering the code.
Exactly. Figuring out the exact language and the mechanism of that cellular listening. And he's not just observing. He's learning the rules and then, learning how to whisper precise instructions or maybe shout commands for therapeutic benefit.
Essentially rewriting parts of the biological instruction manual for health.
Yeah, that's a great way to put it. Deciphering and then judiciously editing life's instructions. Okay, so let's pivot now. Let's talk about one of Garcia's most impactful early contributions, something that really reshaped immunology, visualizing T-cell recognition.
A truly foundational piece of work.
It seems his early work, even back in grad school at Johns Hopkins, was setting the stage. He was looking at how anti-endotypic antibodies recognize peptide antigens.
Which sounds complex.
It is a bit. But basically, these are antibodies that recognize other antibodies, potentially mimicking an antigen's shape. So if an early dive into the principles of molecular recognition, how one molecule specifically finds and binds another.
Laying the groundwork for T cells.
Exactly. Because the pivotal moment came during his postdoc at Scripps. He led this groundbreaking study that revealed, for the very first time, how T cells actually survey peptide fragments presented by MHC proteins on other cells.
The self versus non-self recognition system.
The absolute core of it, distinguishing healthy cells from infected or cancerous ones. And his research led to the monumental first visualization of a T-cell receptor, the TCR, bound to a peptide MHC complex, the PMHC.
Published in Science in 1996.
A huge deal.
Yeah.
It wasn't just an observation. It fundamentally changed how we saw T-cell immunity.
Oh, absolutely. That 96 paper wasn't just another publication. It was like a scientific earthquake. It shifted the entire field. Before that, immunologists had theories, models, lots of indirect evidence, but they lacked the definitive picture. They knew what T cells did.
Kill infected cells, regulate immunity.
Right. But Garcia provided the actual atomic-level snapshot of how they engage with the antigen. It was like having a map of a city but never having seen the actual building.
Ah, okay. This provided the architectural details.
Exactly. The missing link. And the impact. Immediate and profound. It transformed immunology, gave a rational basis for designing vaccines, helped understand autoimmunity, how T cells might mistake self, and crucially, it catalyzed the whole field of immunotherapy, especially for cancer.
Because that relies on directing T cells, right?
Fundamentally. This structure provided the blueprint for how to do that, how to target that interaction. It was literally a blueprint for immune intervention.
It's amazing how one clear image can just change everything. And his work on T cells didn't stop there, did it? when he got to Stanford.
No, his lab continued to push the boundaries. In 2007, for instance, they reported the structure of the pre-B cell receptor, the pre-BCR.
What was significant about that one?
It revealed a completely different mechanism. It showed how these pre-BCRs cluster together oligomerize to signal without needing an antigen.
So not the usual key and lock thing.
Not at all. Sometimes just forming the right cluster is enough to send a powerful signal. It showed this whole other layer of control in how immune cells develop.
And they kept publishing landmark papers in this area.
They did, including the first structure of a co-TCR PMHC complex.
Gamma delta T cells.
Yeah.
They're different, right?
Very distinct, yeah. Different roles, sort of first line of defense in some tissues. Understanding their interactions was a whole new puzzle. They also figured out the molecular basis for how TCRs can recognize both self and foreign MHCs, showing this remarkable adaptability.
Fine-tuned scanning.
And they looked into the germline basis, suggesting maybe innate predispositions in how these interactions happen.
And what about cross-reactivity? I remember reading about that.
Yeah, that was vital work too. Quantifying how one single TCR can actually react to multiple different PMHCs.
Which is good for fighting lots of bugs.
Right, gives you immune breadth. But it's also a major source of autoimmunity, where that TCR mistakenly hits a self-peptide.
The downside of flexibility.
Exactly. And finally, they really nailed down the structural trigger for TCR signaling itself, moving beyond just how they bind to how that binding event actually kicks off the signaling cascade inside the cell.
So from the static picture to the dynamic mechanism.
Precisely. Understanding the conformational change, the flick of the wrist, that sets everything off. And again, this deep foundational knowledge wasn't just for textbooks. It directly fuels new technologies. One of the big recent developments is a peptide MHC library technology.
Okay, what does that let scientists do?
Well, imagine you find a T cell in a tumor that seems to be fighting the cancer, but you don't know what specific target it's recognizing, its antigen. This technology, often using yeast display, lets you create huge libraries of potential peptide MHC complexes.
Like a giant lineup of suspects.
Kind of, yeah. You can screen these libraries to find the specific antigen for those orphan T cell receptors, especially the ones inside tumors, the TILs.
Finding the target for the tumor-fighting T cells.
Exactly. It's a key tool for precision immunotherapy. Understanding what the T cells are trying to fight helps us help them do it better. And, interestingly, this tech also gave them new ways to study the initiation of signaling, refining those earlier structural trigger insights even further.
That's just incredible. It really feels like unlocking a secret code, doesn't it? Being able to direct our own immune system with such precision. Okay, so from the immune system, let's broaden out. Because Garcia's curiosity and his structural toolkit, they weren't just limited to immune cells. He turned that same lens onto other vital cellular communications.
A whole symphony of interactions.
Yeah, exactly. Here's where it gets really captivating, seeing this structural approach applied across different signaling systems. Okay, first up in this symphony, cytokine signaling, the language of cells you called it earlier.
Right. These small proteins are crucial messengers, especially in the immune system, but also in development, repair, all sorts of things.
And Garcia's work established the fundamental principles, right?
Right.
How they bind receptors, how they activate signals.
Yeah, he provided the structural basis for a lot of it. His lab determined the first crystal structures for several key cytokine families bound to their receptors.
Like which ones?
Well, the GP130 family that includes IL-6, a big player in inflammation and development, and the common gamma chain family, IL-2, is in there, vital for T cell growth.
And others too?
Oh yeah, type I and type 3 interferons, critical for fighting viruses. And structures for many others, IL-1, IL-4, IL-13, IL-15, IL-17, IL-23, LIF, CNTF, a huge body of work.
So what did all these different structures actually reveal? Was there one common theme?
That's the interesting part, not really one theme. They showed this incredible diversity in how they bind, different architectures, different topologies. It suggests nature, through convergent evolution, found many different ways for these receptors to get the signal across the membrane.
A masterclass in molecular problem solving, as you said.
Exactly. Lots of ingenious solutions.
And again, it wasn't just about understanding.
No, they leveraged that knowledge. Garcia's group used directed evolution that accelerated evolution in the lab to engineer cytokine variants.
To make them better.
Essentially, yeah. They created versions of IL-2, IL-4, and IFN with much higher affinity, better stability, improved therapeutic properties.
So like designing a super cytokine.
That's the idea.
Yeah.
One that could more effectively boost an immune response against cancer, or maybe calm inflammation in autoimmune disease, but with more precision and fewer side effects. Direct application of that structural insight.
Okay, moving on to another fundamental system, want signaling. You call this one a donut-shaped mystery.
Huh. Yeah, that refers to the structure they found. Want signaling is absolutely crucial for embryonic development, tissue regeneration, maintaining stem cells, really fundamental stuff.
And his lab had a big moment here.
They did, back in 2012. They got the crystal structure of a want protein bound to its receptor frizzled.
And what was so aha about it?
It showed this really unusual binding mode. Want proteins use a lipid modification, a fatty acid chain stuck onto the protein to directly grab onto the frizzled receptor.
That's not typical for soluble ligands.
Not at all. Usually it's protein contact. This lipid key fitting into a specific lock was unexpected, and the overall structure of the complex, it formed this striking donut shape.
Which made the cover of Science.
It did. It completely changed the view of how wants communicate, highlighting this unique lipid-mediated interaction.
In knowing that mechanism, that donut shape, that opened up therapeutic possibilities.
Huge possibilities. More recently, his lab reported they could actually recreate want signaling the main canonical pathway using engineered molecules.
Why did they do that?
They designed these water-soluble bispecific ligands, molecules that grab onto both frizzled and another co-receptor, LRP6, and bring them together, mimicking the natural activation.
And the implications.
Massive for regenerative medicine. Being able to precisely turn on these powerful repair and growth pathways. Think about repairing damaged tissues, maybe even growing new ones. It's a direct result of understanding that initial structure.
All right, next up, notch signaling, another critical pathway for cell fate decisions, development.
And notoriously tricky to study structurally.
But Garcia's lab managed it.
They did. In 2015 and 2017, they published the first atomic-level views of notch signaling complexes in Science.
How do they overcome the difficulties? The interactions are weak, right?
Yeah, low affinity. So they cleverly used directed evolution again to strengthen the binding between Notch1 and its ligands, like delta-like-4, DLL4, and JAG1, JAG1, just enough to stabilize them for crystallization.
Smart workaround. And what did the structures show?
Long, narrow binding interfaces.
Well,
But with a real surprise. These interfaces were heavily decorated with O-linked fucose and glucose modifications on notch. Sugars.
sugar's right at the binding site.
Exactly. And O-linked glycans like that are rarely seen playing such a direct role in protein interfaces. It immediately explained how changing the glycosylation, changing those sugars, could directly tune notch signaling.
An unexpected chemical detail with big consequences.
But wait, there's more. The 2017 paper also showed that these notch-ligand interactions form catch bonds.
Attach bonds. Remind me.
They get stronger under mechanical force, up to a point. Counterintuitive, right? Normally, pull harder, things break faster.
So pulling actually stabilizes the connection.
For a bit, yes. Which raises fascinating questions about how cells physically pull on each other to communicate. They found that delta and jagged ligands have different force thresholds to activate notch.
Suggesting a sophisticated mechanosensing mechanism.
Precisely. Cells aren't just sending chemical signals, they're literally feeling each other out with physical tension translating directly into a biochemical response. The pulling is part of the message.
That is remarkable. A handshake that gets stronger when you pull, revealing how physical forces impact cellular decisions. Amazing stuff. Okay, finally in this section, let's touch on GPCR signaling. G-protein coupled receptors.
Huge class of drug targets. Absolutely massive.
Involved in almost everything, mediating responses to hormones, neurotransmitters, senses, and a huge percentage of drugs target them.
Around 30–40%, yeah. So understanding them is critical for medicine.
And Garcia's lab made breakthroughs here too, especially with protein ligands.
They did. In 2015, they reported the structure of a viral GPCR, US28, bound to its chemokine ligand, fractalkine. Now, visualizing a protein ligand bound to a GPCR, that was a major achievement. GPCRs are tricky enough to crystallize on their own, let alone with big, flexible protein partners.
So what did that structure reveal?
A cool two-part mechanism. The globular head of fractalkine docks onto the GPCR's extracellular loops, but then its flexible N-terminal tail actually threads down into a cavity in the center of the GPCR.
Threading into the receptor.
Yeah, like feeding a string into a hole. And this threading seemed to fine-tune the signaling output, controlling the specific message sent inside the cell.
And this led to deeper insights about GPCR activation in general.
It did. More recent work from the lab, using engineered biased ligands, ones designed to push signaling down one path versus another, showed something really fundamental. It suggested that GPCR activation might be governed less by highly specific chemical bonds, the traditional key and lock idea, and more by ligands inducing specific shape changes or conformational shifts in the receptor.
The dance move idea.
Exactly. It shifts how we think about designing drugs. Maybe it's not just about finding a key that fits, but finding a molecule that makes a receptor adopt the right shape, the right dynamic conformation to get the desired therapeutic effect.
A much more dynamic view of drug design.
And potentially much more powerful for creating specific, effective drugs with fewer side effects.
Okay, so we've seen all this incredible fundamental work, visualizing T cells, decoding cytokines, Wnt, Notch, GPCRs. But what does it all mean for, actual patients?
Right, the translational impact.
These insights aren't just for textbooks. They're fueling new ways to treat really tough diseases. Let's focus on cancer immunotherapy, where his work has clearly had a huge impact.
Definitely a major area. His lab has made key contributions there, turning that structural understanding into potential therapies.
Like the CD47 work.
Exactly. Back in 2013, they developed high-affinity antagonists for CD47. That's the "don't eat me" signal that cancer cells use to hide from the immune system's macrophages.
So these antagonists block that signal.
Right. They essentially unmask the cancer cells, allowing immune cells like macrophages to see them and attack, especially enhancing the effect of other therapeutic antibodies. But then came a really crucial follow-up insight. Garcia's team found that the best therapeutic effects of blocking CD47 actually require combination therapy.
What do you mean?
Meaning you need to combine it with checkpoint blockade antibodies, the ones like anti-PD-1 that take the brakes off T cells, especially in a host with a working immune system.
So CD47 blockade isn't enough on its own.
It seems it works best by stimulating the adaptive immune system, the T cells. It synergizes with the checkpoint blockade. It's not a standalone fix, but a powerful part of a broader immune activation strategy. Connects right back to T-cell biology.
Oh, that makes sense. It's about a multi-pronged attack.
And they've also worked on engineering the immune cells themselves. They created something called an orthogonal IL-2 receptor complex.
Orthogonal.
Meaning it's a synthetic receptor that only responds to a modified synthetic version of IL-2, not the natural kind.
Why is that useful?
It allows selective delivery of that potent IL-2 signal only to the engineered T cells you're using for therapy, like our T cells. Natural IL-2 can cause widespread side effects because it activates lots of cells. This orthogonal system targets the boost just to the cancer-fighting cells.
Minimizing side effects, maximizing the punch.
Precisely. Safer and potentially more effective. And one more thing in this area, their technology using yeast-displayed peptide MHCs to identify tumor antigens recognized by TILs, those tumor-infiltrating lymphocytes.
We touched on this earlier, finding out what the T cells in the tumor are actually targeting.
Exactly. It's like finding the specific flags on the tumor that the patient's own immune system is trying to attack.
Which is like giving the immune cells a highly specialized map, right, to find and destroy the cancer cells more effectively.
That's a great analogy. Identify the unique flags, then engineer T cells or therapies to go straight for those targets, revolutionizing precision immunotherapy.
And beyond the academic lab, Garcia has been directly involved in translating these discoveries through biotech companies.
Yes, he's taken that entrepreneurial step. He's a co-founder of several companies.
Which ones?
Alexa Therapeutics, focusing on immune checkpoints like CD47, Serozin, which is working on regenerative medicines by targeting 1Nth signaling, direct link there, and 3T Biosciences, developing novel T-cell receptor-based immunotherapies, leveraging his deep expertise in TCRs and antigen discovery.
So these companies are directly trying to bring his lab's science to the clinic.
Absolutely. It demonstrates that full circle, from basic structural insight deep in the lab all the way to potential patient benefit. It really underscores his commitment to making sure this knowledge gets applied to improve human health.
Truly inspiring to see that bridge built so effectively, from fundamental discovery to tangible impact. Now, with all this intense scientific work, you might think there's no time for anything else. But let's quickly acknowledge the recognition he's received. It signals the impact, right?
Definitely. He was elected to the National Academy of Sciences in 2012.
And the National Academy of Medicine in 2016.
Prestigious honors, plus the Pisano Award recently in 2024, and earlier awards like Pew Scholar, Keck Distinguished Medical Scholar. Clear signs of his influence.
Yeah, recognized early on for his potential and consistently delivering.
So what's also fascinating, I think, is how that kind of dedication and rigor often shows up elsewhere in life.
Oh.
It takes incredible endurance and focus to do this kind of science for decades, and apparently Garcia channels that same drive into competitive long-distance running.
Really? How
Yeah.
competitive?
Well, according to reports, he's run more than 120 ultramarathons, including several 100-mile races.
100 miles. Wow.
Yeah.
That's unbelievable endurance.
Isn't it?
You know, that discipline, that perseverance, the strategic thinking you need for an ultramarathon, it absolutely resonates with the sustained effort and meticulous planning needed for decades of groundbreaking science.
There's definitely a parallel there, pushing boundaries.
Yeah, pushing boundaries both physically and intellectually.
Yeah.
It's about that relentless drive, overcoming obstacles, whether it's a tough crystallization problem or mile 80 of a race. It kind of encapsulates his whole approach, doesn't it? Hashtag tag outro.
And that really brings us to the end of our deep dive today into the incredible career of K. Christopher Garcia. We've journeyed through this amazing molecular world, seeing how he uniquely blends structural biology, biochemistry, protein engineering. His genius seems to be in visualizing and then truly understanding how cells communicate at the most fundamental level.
From those first pictures of TCRs engaging antigens, which changed immunology, right, to decoding cytokines, Wnt/Notch, GPCRs, his work hasn't just advanced fields, it's genuinely transformed our understanding of immunity development disease. And it's paved the way for therapies that are already making a difference.
You know, it raises that important question about the ultimate value of structural biology. When you can see the architecture of these interactions, understand the shapes, how they fit, how they move, you gain the power not just to understand disease, but to rationally engineer solutions.
Moving from just observing to actually designing.
Exactly. It's molecular design at its most fundamental level, driven by deep insight into life's machinery. It's the blueprint for building the medicines of the future.
Absolutely. So, as you go about your day, maybe just take a second to consider that almost unimaginable complexity inside every single cell in your body. And think about how understanding these fundamental interactions like Garcia has done isn't just intellectually satisfying. It unlocks possibilities for health and healing we couldn't have dreamed of before. It turns mysteries into solvable problems. So, how much more deep diving is left for us to do? What other hidden structures, what other molecular dances are just waiting to be revealed, holding the keys to future cures? Or maybe just a deeper appreciation for life itself? Thank you for joining us on The Deep Dive.